my name is Devin Forcina. I'm the Communications Specialist at the Irving Institute for Cancer Dynamics at Columbia University, and welcome back to our Faces of IICD video series. Today I'm here with Dr. Genevera Allen.
I'm Genevera Allen. I am a professor in the Department of Statistics, and I'm also an investigator at the Zuckerman Institute for Mind, Brain, and Behavior, as well as, of course, the Irving Institute for Cancer Dynamics.
So why are you excited to be affiliated with IICD?
Great, yeah. IICD is a great hub on campus for computational biology work, especially as it relates to cancer. And I'm a statistician, and what I do in my research is develop new statistical tools to help scientists make reliable and reproducible discoveries from their data. This is critical when it comes to computational biology, and especially genomics, where there's a ton of new technologies to produce all sorts of high throughput data, and scientists need to get in there and make some discoveries so they can understand how cancer arises, how it evolves, and potentially therapies for cancer. So what my group is developing right now is new frameworks to do uncertainty quantification for the results of unsupervised analysis that we can apply to single-cell genomics data. So I'm developing methods to directly test the uncertainty or quantify the uncertainty in those manifold embeddings. And further, often people then cluster the data into cell types. Say a very simple cell type is to distinguish between tumor and non-tumor cells and find those cell types. And we often do that via methods called clustering or finding groups in the data. And also we're trying to say, hey, what's the uncertainty in the cluster labels? When we cluster and we say, hey, this cell is a tumor cell and this cell is a normal cell, how certain are we in that cluster assignment? Is there some uncertainty there? And this is where kind of statistics meets machine learning meets biology.
Shifting gears a little bit, you were mentioning your group a lot.
Yes.
Tell me a little bit about working with your group and your mentoring philosophy there.
Yeah, so my group consists of one or two postdocs, a handful of graduate students, and I usually work with some master's or undergrad students. I've also certainly worked with REU students from the IICD REU program over the summers as well. In my group, each student kind of has a major focus area that they're working on in their research, but we certainly have major themes and collaborative projects amongst the group. Some of my students also collaborate directly with scientists and help them to work with their real genomics data to make sense of this and analyze that data for their specific problems.
What advice would you have for young up-and-coming scientists?
What advice? So I'm going to restrict this to up-and-coming scientists, focusing more on kind of the computational biology side, and specifically statistics, machine learning meets biology. I would say immerse yourself in real problems and real data, right? Don't just restrict yourself to learning computer science and pure math and maybe some statistics. You've got to get your hands on real data sets. If you're interested in getting into computational biology, get into some of those real data sets. Understand how big they are, how complex they are. What are standard pipelines and workflows that people use to analyze that data? And that's critical. So in addition to kind of the core technical skill sets in math and computer science that you need, I think it's critical as you're starting out to get experience working with real data, and also experience working with scientists and how to communicate with them and explain what you're doing and understand their scientific goals as well.
I know that when you were with Rice University, you developed a data science education center there, and have always been active in creating centers for education throughout your work. Tell me a little bit more about your career journey, like mentors that have shaped you along the way, pivotal moments.
Okay, career journey. Okay, so I think I'm going to go back to grad school, which was a while ago for me at this point. But so I would say in grad school, my advisor, Rob Tibshirani, was really, I think his influence on me was really pivotal in his encouragement to work on real problems that matter to scientists, and not just to work on stylized like math problems or theoretical problems, but actually to try to find specific statistical and machine learning problems that can make an impact in the real world. Mm-hmm. And so I think he really influenced me a lot in that respect, and I think everything that I've tried to do since then, I really focus on how can I develop techniques that can be used by real scientists in practice. And so I went to grad school at Stanford. I started as a professor at Rice and Baylor College of Medicine. And along the way, somewhere along there, I actually founded and then became the director of a data science center. You alluded to this. And so this data science center started some of the data science degree programs at Rice. And a big thing we started was an introductory data science program and also a capstone program in data science. And there are lots of other activities around kind of getting students involved in data science at Rice. I would say it was a lot of work doing that. It was fun, especially founding things and setting things up. But I think in moving to Columbia 2 years ago, my goal was really to get back to focusing on research.
While you've been at Columbia, you work with the Center for Theoretical Neuroscience, obviously IICD. What's it like collaborating across different groups or centers?
Yeah, so I definitely have several affiliations, and I actually think that's a huge plus and a big bonus, because with what I do, say I'm trying to do uncertainty quantification for machine learning as applied to science. This is applicable in genomics and computational biology, but it's totally applicable in neuroscience too. People need to cluster their big neural recording data to discover neural cell types. And now they're actually getting multimodal data where they've got this single cell genomics data plus neural recording data plus maybe animal behavior data, and they want to put it all together and apply these machine learning techniques. So a lot of the machine learning techniques I develop can be totally used in many different domains.
Is a fun fact or something about you that you would want to share with people watching?
Okay, fun fact. Let's see. Well, I really enjoy hiking a lot. In fact, I live north of the city, not in the city, so I can go hiking out my backyard, which I love, which I do with my two dogs, who are basset hounds. Basset hounds, the big, long, dangly, floppy ears, they're a mess, but surprisingly, they're great hikers, and they love to hike with me, so it's great. And I try to get out on the weekends, but sometimes even, on a quick break during the week, and get out hiking.
Thank you for everyone watching, and stay tuned for our next Faces of IICD.